Okay, hello everyone. I think we can slowly start. Before we start, I think we don't have many announcements, but we'll be back next year. So enjoy Christmas, enjoy holidays, and see you next year. I think most of you or all of you should get email from Elena Rivas. So Elena Rivas and Eric Veskov are organizing workshop in Benasque. It's very different from anything else probably that you've seen before. It's 2 weeks in Spain and plenty of time to discuss. Lectures or organized meetings are relatively short, and there is plenty of time to collaborate with people. So if you want to learn more, ask me or Elena or Eric Veskov. They are the main organizers. And I think that's all for today in terms of announcements. And now I would like to introduce our speaker, who is Miroslav. I was happy to collaborate with Miroslav or talked about projects, and I know he also collaborated with other people here. So he did his PhD at Masaryk University in 2007, and then right after that he obtained permanent research position, which is very comfortable, at the Institute of Biophysics of Czech Academy of Sciences in Brno. Currently, he's a senior researcher there with over 40 publications and very interesting collaboration with some of the people that for sure I know, like Marcin Nowotny from IMCB. This is where I did my PhD in Poland, and also Petr Schulz, ISU in Arizona. And his area of research is simulations of theoretical studies of nucleic acid and proteins. And however, he has a very long track record of successful collaborations with experimentalists. And right now, his main focus is to learn about the role of dynamics in biomolecular system and how it's utilized in evolution. So with that, Miroslav, I'm really happy to hear your talk, and everything should be working. No. But the video, sorry guys. Hopefully the video will be back, but we had some problem with the video, but the voice should work.
Okay, yes. So I hope everyone can hear me. Thank you, Marcin, for very nice introduction. So I will go straight to my talk. Okay, so as Marcin already mentioned some of it, my main method of research are molecular dynamics simulations. And the topic I'm interested in include RNA structural dynamics and folding, especially of non-canonical motifs. Basically, the weirder the RNA is, the more I enjoy researching it. I also study non-canonical DNAs, such as the holiday junction, but my main focus are protein RNA complexes. And of course, I do the associated method development with all of these topics. So first, just a basic question, some description of molecular dynamics. So it's a method for describing atomic motion using the classical Newtonian mechanics. For this reason, it's often called ball and spring approximation, because basically you can imagine the atoms connected by springs and the interactions between them facilitated by the stiffness of these springs and their length and so on. So of course the quality of the molecular dynamic simulations are limited by the underlying model, which is called force field. And the key question is, what should the ball and the spring behave like in order to approximate the atomic motion of the real biomolecule? So why are we even using such simple basic approximation? The reason is that it provides structural details and time resolution which is inaccessible for the experimental methods. And compared to full quantum chemical calculations, which would include electronic structure effects, it's computationally very cheap, so it allows us to run long simulations. Of course, the force field, the empirical nature of the force field isn't the only limitation. Another limitation is a typical setup of the molecular dynamic simulation. where you have the solute molecule you are studying surrounded in a box of water molecules. And one of the problems is that in order to keep the computational costs acceptable, the size of this box is often very small. In fact, if you had such concentration of solute in experiments, it would immediately lead to coagulation. So the concentration of solutes is very high, and of course the ion concentration is also very high. Nevertheless, big advantage of molecular dynamics is that they are basically a hypothetical single molecule experiment you would obtain with optical microscope and with infinite resolution. So it's something which is impossible to have experimentally, and there is no equivalent experimental method to obtain the data you can obtain with molecular dynamic simulations. So often when I collaborate with experimentalists, they can have, at least in the beginning, this impression of computational chemistry. They can think that it's like I'm press the button and then we get a great paper. So it's not like this, unfortunately. More often it's like this. I can take some structure of protein RNA complex from the database, and then I can observe that it gradually disintegrates in the simulation, and in the end it completely collapses. So do I get some nice paper like this? No. It ends up in a garbage. So this is more common than you might think. In fact, the problems with simulations are discovered all the time as the computers get better, and we can get to longer time scales. If I were to evaluate my career so far, then I would say that I have spent 50% of my time trying to perfect the methodology and 50% of time on answering the actual chemical and biological questions using this methodology. Of course, when you try to do method development, you must be very careful to avoid overfitting for one particular system. Often what people do is that they see one problem is in a single system, for example, in this protein RNA complex, and then they try to fix the force field, they try to fit it to reproduce this system exactly. They can do that, it's relatively straightforward, but then you try to use this improved model in another system, and it's a complete disaster. So you must be careful with this to avoid overfitting, or it's better not to claim general applicability of such method. So since you can encounter problems in a single system, it's a very good practice when you are doing simulations to have a diverse portfolio of various molecular systems. Then, if you encounter problems in one of them, you still have the other systems to work with. But even then, there are certain best practices in molecular dynamics that is good to follow. Basically, you need to ask yourself several questions. The first one is your starting structure in the database? Do I have something to work with? Usually, it's much better to have some experimental structure than trying to model something from the scratch. And then if you have the experimental structure, is it of sufficient quality? Isn't there, for example, some crystal packing or something like that? And then even if you have a perfect experimental structure, you need to ask yourself if your simulation is correctly executed. There are several steps you need to follow. And then finally, if you really executed the simulation correctly, the final question is your simulation giving you a sensible result? The problem with computational chemistry is that the computers always give you a number. They always give you a result, and then it's up to you to decide if that result makes any sense. So if you are lucky and the
Miroslav,
answer,
can I ask a question here,
yes.
maybe? Because you mentioned it's better to start with the structure, like the experimental structures done with the like with modeling, like models from the modeling. But with the AlphaFold, what is your opinion about using AlphaFold models? And have you seen like people using AlphaFold models for molecular dynamics? And are there any specific problems with the AlphaFold models, for example, in terms of at least in proteins in your work?
Now, I think the main problem with using the models isn't so much with the model itself, but with the sampling limitations of the simulations. And the problem is that any model compared to experimental structure, even if you have a very good model, there will be a slight inaccuracy compared to what you would obtain with the experiment. And then if you try to use this model in simulations, it's very likely that you won't be able to correct this inaccuracy on your simulation timescale. So yes, so even assuming you have a very good model with very low inaccuracy, you will encounter this problem. So for this reason, I always prefer working with the experimental structures.
Thank you.
Okay, so as I said, if you are lucky and everything is okay with your simulation, then there are many interesting scientific questions you can answer using the simulations, and one of them is the structural role of solvent, of waters and ions for the stability of the RNA structure. The thing is that the nucleic acids, they are polyanionic, so it means they are strongly attractive for waters and cations. The solvent is part of what stabilizes the nucleic acids. But at the same time, the ions and the waters, there is so many of them in the system, and they are moving really fast, and they are small. So it's difficult to see them with static ensemble averaged experimental methods such as the X-ray crystallography. So this is one of the areas where molecular dynamics can really help, because in molecular dynamics you can always see the position of every single molecule of water, of every single ion. And we applied this in the past, for example, to learn of a particular monovalent ion binding site in this ribose switch. More specifically, it's U-turn motif, which caps this motif. I'm not sure if you can see my pointer or no. Can you see my pointer?
Yeah, we can see.
Okay, great. Okay, great. So we observed using the simulations that there is this very strong ion binding site at this location, and this was subsequently confirmed using the NMR experiment. You can see the NMR spectra here. Don't ask me how, but it somehow confirms the observation of this ion binding site. And what was very interesting for me is that this ion binding site is actually a subject of evolution. Because when we looked at the U-turn motif in the structural database, and we looked at these other structures, we observed that the uracil at this position can be substituted with protonated cytosine. And basically, what this corresponds to is this potassium, this positive charge coming from the potassium, being integrated into the nucleotide. So one of the things that we concluded from this research is that the solvent interactions are actually a subject of evolution. So another thing that we can learn using molecular dynamics are the rare conformational states in biomolecules. The thing with experimental structures using the structural biology methods like X-ray crystallography, cryo-EM, or NMR is that they provide a static ensemble average structural model, which means that the model you see it corresponds to a structure in equilibrium. But what can often happen with biomolecules is that the rare, or you can also say transient conformation, is the one that is having some biological role, some biological effect. So what we can do using molecular dynamics is that we can take the structure in equilibrium that was obtained using the experiments, and we can use molecular dynamics to observe these rare conformations. And this is what we did in this structure. This is structure of a RAFC enzyme binding to holiday junction. The RAFC is an enzyme which is responsible for the resolution of the holiday junction, and by resolution I mean the separation of the two joint DNA molecules into back into the two separate molecules. And what we observed, or what was known before, is that the RAFC binds any sequence. It binds any holiday junction. It doesn't matter what the sequence is, but it is capable of cleaving, of separating only holiday junctions of specific sequences. So what we did is that we performed simulation of this RAFC bound to holiday junction using different sequences, and we observed that there is a very specific conformational state consisting of base flipping, which is accessible only for the cognate sequence, only for the sequence which is being cleaved. So just to summarize, what happens is that the RAFC binds holiday junction. It binds any holiday junction, but it binds it in inactive geometry, which is it's in geometry which precludes the cleaving. So what then happens is that the RAFC is capable of disrupting a single base pair, but only in the cognate DNA sequence. It facilitates this via base flipping, and after the enzyme does this, it can assume the catalytic geometry and actually perform the cleavage. And we also observed that the RAFC, using the same structural mechanism, it's capable of stabilizing. It's capable of preventing quick reformation of this base pair, and so it not only facilitates the catalytic geometry, it also stabilizes it and promotes it in the ensemble. So yes, can you see the video?
Yes.
Okay, so good. Videos usually don't work in presentation, so I'm very happy that this is not the case here. And what you can see in this video is this arginine of the RAFC. It constantly samples this base pair, and eventually it helps in its disintegration. And then it starts with interacting with this adenine, preventing it from going back to interaction with this thymine. And this allows this side of the molecule to go to the side and start interacting with the catalytic side of the RAFC. So, and this is highly specific for this cognate sequence, because if there was a GC or CG, then the RAFC would be incapable of separating the base pair because it's too strong. And if there was AT instead of TA, then the arginine would be incapable of forming this interaction with the thymine. So this is highly specific for the TA, and it's all being accomplished by this rare conformation, by this transient state. So naturally, after this project, we basically fell in love with the holiday junctions. They are not only biologically interesting, but they also have a nanotechnological applications. Namely, they can be used as a repeating unit for construction of these beautiful crystal cells. And the idea behind DNA nanotechnology is that you can construct cavities with a certain size and properties, and then you can use them as containers for isolating other molecules. So you can use them as a programmable containers for other molecules. And what we did using the simulations is that we performed simulations of all 36 sequences of holiday junctions which can be used to construct these lattices. Because what was observed is that some sequences of holiday junctions are incapable of forming these crystals, or they are forming the crystal really poorly. And we rationalize this observation using the simulations by showing that the sequences which are not capable of crystallizations have a very high dynamics, and they also lack a very specific ion binding properties. So using this research, we were able to recommend what sequences of holiday junctions should be avoided, and which sequences should be preferred when constructing these 3D lattices. So this was just a short sidetrack concerning the protein DNA, but my main area of research are protein RNA, or protein nucleic complexes in general. The thing is that the proteins are a very important player in the metabolism of nucleic acids. And the reason is that the DNA, it's almost always, and the RNA, it always interacts with the proteins in living cells in vivo. In fact, if you have an isolated RNA, then the first reaction of any cell is to immediately destroy it, because it assumes that it's some kind of virus. So when you are talking about the metabolism of or about the biological function of the RNA, then it's important not to omit its interaction with proteins. Yeah, the proteins handle every aspect of the metabolism of nucleic acids. So when you want to do simulations of protein nucleic acid complexes, such simulation is of course more complicated than when doing simulations of the isolated biopolymers. And the reason is that you have to use two different force fields, one for protein and one for nucleic acid. So it means that you have a double chance for something going wrong, yeah, for double chance for some error in the force field. Another problem with the protein and nucleic acid simulations is the relative lack of initial structures, because often in the structural database you have only the structure of the isolated biomolecules. Even if you have a good structure to start with, you can still encounter unique simulation and force field problems which are unique to the protein RNA interface, which you won't encounter in the isolated biomolecules. So one of the basic questions, which force field to use, it's a good practice to start with the state-of-the-art force fields for the nucleic acids and for the protein. But still, you don't know in advance how the two force fields will behave in the simulation of the protein nucleic acid complex. So can you tell somehow in advance if a specific protein RNA complex is going to be stable in molecular dynamics? So the answer is that you can't, because unfortunately what we observed in the past is a strongly system-dependent performance. We observed structures for which the simulations gave an entirely stable performance. We observed structures where we had some trajectories which were stable and some were unstable. And finally, we had systems where we could not obtain a stable trajectory no matter what we tried. So whenever you are doing simulations of protein nucleic acid complexes, you need to do preliminary set of simulations and see how your system behaves. It's a good idea to do that before you invest more computational time into it. But if you're lucky and your simulation of the complexes works well, there are again various interesting biological and biochemical aspects of the system you can study, such as the interface conformational dynamics. You can see the fast movements of the protein RNA interface, which again you can't see in static experiments. You can use the simulations to deconstruct the structure averaging. I will explain in a moment what it is. You can also use it to study hydration, which I already mentioned is a strong application of molecular dynamics. And relatively recently, we managed to study the the details of the binding mechanisms for some selected protein RNA complexes. So in most of my studies, I focused on one particular protein motif, which is binding RNA in living cells, and it's the RNA binding RNA binding motif, which is abbreviated as RRM. And this protein motif possesses a relatively simple topology of beta alpha beta beta alpha beta, but it's capable of binding a great many different RNA sequences by small variations in its surface exposed amino acids. It's very versatile, and using this simple motif, the proteins can be tuned to binding basically arbitrary RNA sequence. At the same time, the RNA domains have a very long history with molecular dynamics. I mean, the first simulations of the U1A RNA, they are already from 1999, so it's a very long history. This U1A RNA complex, it was studied in the past a lot. And there is a reason for it, and the reason is that it possesses a relatively rigid interface and very strong affinity, which is unusual among RNAs. But because it's so rigid and so strong affinity, people were able to observe stable simulations even with relatively old force fields. But nowadays, even for this very stable system, we can see performance problem already on microsecond time scale. So you can imagine that when the other RMs have a lot more dynamics and their binding isn't so strong, then we observe a lot more problems in these simulations than in the simulations of the U1A. But again, if you are lucky and your simulations are giving good results, then you can study various things, and one of them is this structure averaging I already mentioned. We studied this in the FOX1 complex. FOX1 is it's binding the RNA using the RNA domain, and so just to explain what is structure averaging in experimental structures. So structure averaging occurs when you have an experimental method and you are trying to observe a molecular movement, and this molecular movement is faster than is the time resolution of your experimental method. So when that happens, two things can be the result. Either you observe, if you imagine this simplified model, if you imagine a single particle moving in a spherical region, what can happen is that in case of structure averaging, what can happen is that either you can obtain a completely blurry picture, so the only thing you can tell for certain that the particle is moving somewhere within the sphere, or the second option that can occur is that you obtain a structure average structure, and you observe that the particle is exactly in the center of the sphere. So if this happens, and it happens in the biomolecular structure, what we can do with simulations is that we take such structure and we reconstruct the movement of the particle of the biomolecule starting from the structure average structure. So this is what we did for this system. What we observed using the simulation is that there is this particular arrangement of nucleotides and amino acids, which is forming a so-called hydrophobic pocket. In essence, you have 3 different nucleotides wrapped around a single phenylalanine side chain. And what we observed in the simulations is that this hydrophobic pocket can exist as 3 different structural arrangements, which are exchanging along the simulation trajectory. And what was very interesting is then when we compared such trajectories against the experimental data, we had an agreement. So when we combine the populations, all of these conformations, it's actually in the agreement with the experimental data. So our conclusion was that this conformation can actually exist in the real molecular ensemble, but in the experimental structure they average into a single conformation. So we can use simulations to uncover cases like this. So another strong application of MD I already mentioned is the hydration. So what we observed in the simulations, we observed several structurally important hydration sites where the water molecule, single water molecule or fast exchanges water molecules are always present. And what we then did is that or rather our experimental collaborators, they prepared mutated proteins where these hydration sites are disrupted. For example, this one, they mutated this serine into alanine. And what we observed, again using the experiments, is that when this is mutated into alanine, the RNA affinity of such mutated protein goes down. So these hydration sites, they are actually important for the RNA binding affinity of the protein. So yeah, you can imagine in the context of the protein RNA complexes, the water is very more important. You can imagine if you imagine the proteins and RNAs as individual bricks of the wall, then the water is the mortar between the bricks that keeps the whole thing together. So imagine what would happen if you took protein RNA complex and you completely disregarded the water. Then you would be getting completely skewed results. So as I said, the study of hydration, it's a strong application of molecular dynamics because you have only limited information. In X-ray structures, you get average picture. In NMR, you typically can't see water molecules at all. On the other hand, using simulations, you can study position of every single water molecule at any time. And the good thing is that the timescales which are relevant for water dynamics are well accessible in MD, and they have been for some time. Of course, there is also a problem, and the problem is that the study of hydration using simulations is very computationally and personally demanding, especially nowadays when you have a very long simulations, it can get actually problematic just to open the trajectory and view the water molecules. Because it's so big. So when you have to do it, it's very demanding and very tedious. In fact, if you ever want to punish your PhD students or postdocs, just have them analyze hydration in molecular dynamics. That works every time. But if you actually make the effort, you can learn many interesting things. Another.
Miroslav, can I just
Yes?
a question before
Okay, sure, yeah.
you go in the system as before, but yeah, maybe this is not relevant. So the system, or like even here, I think so when you insert water, I think you don't take any positions of water from the experimental structures. Maybe there are some.
Okay,
so
some. Yeah. Okay, sorry, yeah, no, go on. Sorry.
So yeah, so you kind of random, like I think in random way you put water around the molecule, but is there any way to put like water randomly in between the interface, or you just wait till this water will come from outside between the interface?
Okay, so usually it comes from the outside.
Okay.
In the beginning of the simulations, we always do this equilibration period where the solute is gradually unfrozen, and it gives the water molecules time to come inside these pockets. But sometimes it can happen that the water binding site is completely isolated, it's buried within the interface, and then in that case it might be possible that it won't come inside in time. So in that case, it's good to examine the starting structure and see if there are some cavities in the interface where the water molecules might fit. But it's quite rare because these buried water molecules, they are there is big entropic penalty for such buried water molecules. So usually in 90% of cases, the water molecule will come in on its own.
Okay, thank you.
Yes, you're welcome. So yeah, this is just another study where we observed important role of hydration. This is a so-called YTH domain, and this protein it's capable of recognizing the M6 adenosine modification in RNA. And what we observed is that it accomplishes this. It's capable of distinguishing the methylated and unmethylated substrate because the unmethylated substrate is unable to displace the water molecules within the binding pocket for this nucleotide. And at the same time, the water molecules, they are also capable of rapidly displacing the unmethylated substrate from the pocket. So again, the hydration dynamics, the water dynamics, it's capable of both distinguishing facilitating the specificity for the correct substrate, and it's also capable of the dynamics of the water is capable of facilitating the rapid turnover of the incorrect substrate. So right now I mentioned that the things that sort of worked in the past for us, but not everything is so great. And now I would like to share with you one story about something that haven't been working for a long time, and we finally managed to get it work quite recently. So just a brief introduction. In the past there was a very big problem with the simulations, with nucleic acid simulations in general, and that's the RNA overcompaction. So what happens in case of the protein RNA complexes is that you start with the complex from the database, and then you observe your interface gradually disintegrating, and the RNA chain bending over and starting to form RNA interactions instead of the protein RNA interactions. So it was clearly observed in this system, but it can be seen basically in all protein nucleic acid systems. And this problem it's related to the well-known and notorious difficulties with describing the single-stranded RNA molecules in molecular dynamics. Yeah, there are many studies which explore the performance of the simulations for the tetranucleotides. And what usually happens in those simulations is that instead of existing as an extended chain, the RNA molecule tends to bend on itself, and instead of sampling the extended conformation, it instead forms this compacted form, which I am calling RNA globule. And once this happens, once the simulations forms this, it's usually permanent. It's highly stable, and then the RNA tends not to sample anything else. So of course, this is a problem in simulation of isolated RNA, but the same problem exists also in the simulations in complexes. Because when this happens in the simulations, is that the RNA also starts to bend over and starts to interact with itself. What happens is that the RNA interactions, they gradually overpower all other intermolecular contacts. So what happens is that the RNA globule you can see here, it forms also in these systems. And when this happens in the complex, it happens at the expense of the protein RNA interface. And as I said, all RNA force fields, they are all affected. So the best case scenario is that it won't happen on the simulation timescale when you do the simulations of the complexes. So here there are some, there is a picture of this happening in one of the systems. And here there are some graphs. So this is the solvent accessible surface. So the blue curves, they are the standard simulation using the standard force field. Yeah, I will explain the red and green one in a moment. So what happens in the standard force field is the dissolvent accessible surface of the RNA gradually goes down, and this is associated with the disruption of the system and also with the reduction of the protein RNA interface size compared to the experimental value. Right? As you can see, it's a quite large reduction, and it doesn't stop there. It will just keep happening. So as I said, this RNA overcompaction is a big problem because it disrupts the protein RNA interfaces of fully formed complexes. It can, in principle, it can be slowed by stabilizing the native protein RNA interactions using, for example, the HB fixed potential. This is a solution which we have used in the past, and it works because ultimately it just delays the problem. Because the problem comes from the overstabilized RNA-RNA interactions. So this is a problem because it prevents the studies of, for example, of protein RNA binding, because before the RNA has a chance to bind to the protein, it instead forms this hyperstable globule. And it also precludes us from studying large-scale conformational changes, because once the RNA temporarily separates from the protein, it doesn't go back, it instead forms this globule. So, well, what we figured is that in many protein RNA complexes, you have a single-stranded RNA binding to the protein. This is very typical, for example, for the RNM, which I mentioned. So the RNA in such complex, it's forming this extended conformation. And at the same time, there's over a decade of trying to fix this problem. So it's a very difficult problem. It's an uphill battle. So what we figured is that why not just get rid of all of these RNA-RNA interactions? And that's exactly what we did. We prepared system-specific modifications for studies of protein RNA complexes where the RNA is bound as a single strand. So what we did is that we weakened the RNA-RNA interactions using the non-bodied fix, which is basically the modification of specific Lennard-Jones pairwise potentials between selected atoms. We call this modification STEFIX, which is abbreviation of stacking fix. Because in the beginning, we figured that the biggest problem with the RNA compaction is the overstabilized base-base interactions, the stacking. But once we did this, we observed that once we weakened only the stacking, we observed that the base sugar and the base phosphate interactions is also overstabilized. So in the end, we included almost the entire nucleotide in this STEFIX potential. So I would like to stress that this is not a general modification. This is just something which will allow you to study the single-stranded RNAs bound to proteins. If you try to apply this for structured RNAs, then of course your molecule will disintegrate. So it's not a general modification which should be used everywhere. So what we tested it on are these two protein RNA complexes. Both of them are RRMs, but they have a very different characteristic of the protein RNA interface. So we have the binding pockets. In this one, we have the 4 binding pockets, and 2 of them are binding uracils, and 2 of them can also recognize adenines. Meanwhile, this protein has a 3 binding pockets, and it's recognizing just these GGA motifs. So they are very difficult. They are very different. The interfaces are very different, and they also have a different dynamics. But what we observed is that the application of STEFIX it prevents this globule formation in both of them. Yeah. So I think I'm running out of time. So I will just say this fast. So these 2 curves, the red one and the green one, they are from simulations where we applied STEFIX. So what we observed is that when we apply STEFIX, the protein RNA interface is no longer being disrupted, or at least it's a day and night difference compared to the unmodified force field. Yes. So here you can see the occupancy of the nucleotides in the individual binding pockets. So with the standard force field, these two pockets are gradually lost, and they are lost permanently. Meanwhile, with the Steffi's modification, we can keep the complex stable on a 10 microsecond time scale. So we observed a similar story for the other complex. Here, what happens is that the pockets 3 and 2, there is a lot of dynamics with them, and once they temporarily lose the contact with the protein, the RNA globule starts forming with the standard force field. But again, this can be prevented by application of Steffi's, and in this case, we don't observe anymore the disintegration of the protein-RNA interface. It's the structure is stable throughout the simulation. So using this modification, we managed to obtain a more extended conformation of the RNA in simulations. And when we observed that, we also tested this on the structure of isolated RNAs. And what we observed is that we are actually capable of sampling the conformation corresponding to the RNA-bound conformation. So since this could in principle allow the conformational capture mechanism, what we ask ourselves is if we can actually observe the spontaneous RNA binding using simulations. And the answer is that yes, we can. We started with a system where we separated the RNA molecule from the protein by at least 20 angstroms, and then when we performed the simulation, and what we observed that when we use Steffi's, the RNA was actually capable of spontaneously finding its binding site on the protein and assuming the correct binding conformation within the simulation time. It should be stressed that not every simulation was successful, because this is we didn't use any guiding potential, so it was a completely spontaneous process and therefore subject to random sampling. But compared to simulations with the standard force field where we haven't applied Steffi's, in those simulations we were basically incapable of observing the protein RNA binding at all. Yeah, we observed some attempts by the system to form the protein RNA interface, but the system was unable to outcompete these overstabilized RNA-RNA interactions which form the RNA globule. So yeah, using the Steffi's simulations, we were, for example, for these systems, we were capable of observing successful binding in 18 out of 24 simulations. While without Steffi's, the success rate was much lower. For some of the systems, we couldn't observe any successful binding at all. And one of the interesting biological conclusions is that we observed every binding event which we observed in those simulations was different. But at the same time, there were some common things among the binding events which allowed us to form this scheme for the RNA binding for this particular system. So what happened in the case of the HUR RNA3 is that starting from completely unbound biomolecules, we observed the initial contact which happened by one of the uracils binding in one of the pockets. And then we observed a formation of this pre-binding state where the RNA was extensively but still non-specifically interacting with the protein. And finally, the RNA could bind in one of these four binding registers. We call them non-optional or optional. The difference is that in the non-optional binding, only 3 pockets are occupied, while in the optimal binding, you have all 4 pockets occupied. And the interesting thing we observed in our simulations is that the system can transition from this non-optimal binding to the optimal binding, but it does so directly, but rather by temporarily reverting into this pre-binding state where it is very flexible, and then going to the optimal binding. So, and we theorized that the existence of this pre-binding state is how this protein could be tuned to really fast to find the cognate sequence, the sequence that it recognizes really fast. Yeah, because otherwise the exchange would be too slow. In
Miroslav,
contrast,
can you go back quickly
yes, of
to the
course.
previous slide? Can you calculate like bounding energies between these interfaces, and can you see there is some of them in terms of like the energy calculations are more preferable?
Okay, so we haven't tried this, and one of the reasons is that we still had like a insufficient sampling. Yeah, because usually when you do free energy calculations, you calculate the potential energy, and then you somehow include the entropy, and the second part, the inclusion of entropy, it's problematic. So yeah, so we haven't tried this, no.
Okay, thank you.
Yes, welcome. Just I will briefly mention the second complex. It had a very different binding mechanism. Namely, there was no pre-binding state. What we observed for this SRSF1 RRM2 complex is that the RNA simply bound in one pocket, and then in the other, and then finally in the third pocket. So because of this, because there was no pre-binding state which could facilitate the register exchange, the success rate in this system was much lower. The binding was slower, because what could happen is that, for example, the G3 nucleotide would bind in pocket 1, and the only way how to resolve this situation without the pre-binding state was for the RNA to completely separate from the protein and make a completely new binding attempt. And this was a very slow process. So we figured that some of the proteins could be really tuned for this fast turnover, for fast location of the correct sequence, and some of them could be using a different mechanism which is slower. Using this Stefik modification, we managed to overcome this RNA overcompaction problem. It's, as I already mentioned, it's a problem which is present in all Embr variants of the force field. So of course the Stefiks can be combined with any of the Embr nucleic acid force fields. And it allows us to simulate the protein RNA binding. So one of the things I would like to do in the future is to study more binding mechanisms of other RNAs and also other protein RNA complexes. Based on the observation of these two cases, it's quite possible that they could all be unique in their details. So I would like to study this. I would also like to try my hand at the protein-RNA interface prediction, because there are many structures of the proteins in the database which are known to bind nucleic acid, but there is no structure of the complex available. And I would also like to use this method for improved description of the interface dynamics when you simulate already fully formed protein-RNA complexes. And I would also like to explore possible application for the stem-looped RNA and not just the single-stranded RNA, because the stem-looped RNA is also often bound by the proteins. So at the end, I would like to thank the members of my group, past and present. And I would also like to thank my experimental collaborators. And last but not least, I would like to thank the taxpayers from the Czech Republic and European Union for generously supporting my research, and you for your attention.
Thank you very much. Do we have any questions from the audience?
Hello.
Hello.
Yes,
Go ahead.
This is Emily from NCI NIH. I have a quick question about the RRM. So is this protein domain, it's across conserved across all different species, like in virus, bacteria, and human, or it's on I mean eukaryotes, or it's depending on the different species?
Okay, so it's in eukaryotes.
It's only in eukaryotes.
Yes, and
I see.
it's highly conserved. This scaffold is it's highly conserved. And I didn't mention is another thing how it achieves the ability to bind virtually any RNA sequence is that it can utilize any parts of the domain to perform this binding. So the most commonly the RNA binds to the beta sheet surface, but it can also bind to these alpha helices. It can also bind to the RNA loops. So it's a really versatile motif, and it's hyper optimized for binding of nucleic acids. In fact, if you study structure of the proteins or the sequence of the proteins, then one of the sure ways how to tell that the protein is binding nucleic acid is if this domain is present.
So this domain, but you said it's only in human, or I mean, let's say bacteria. If there is, do they also have this domain, or they have a different domain to bind bacterial RNAs?
Oh, to be honest, I think this is only in eukaryotes, but I don't want to be misleading you. I think so far what every RNA we studied was from eukaryotes. So, and I think it's only in eukaryotes. I mean, the metabolism of the RNA in bacteria is a little different. The genetic expression is much more straightforward there, and the existence of the RNA is more short-lived. So there is a different set of tools that the bacteria is utilizing to metabolize the RNA. While in eukaryotes, you have the RNA splicing, which is really complicated, and the metabolism of the RNA is much more complicated. So the set of tools utilized by eukaryotes, it's much more rich when it comes to metabolism of the RNA.
So how about virus? Do you know whether virus protein, let's say like infect a human, do they have this kind of domain?
I don't think they do.
Okay, all right.
Yeah, I just Googled and checked, and also Janusz Bujnicki added the paper. So yeah, they are RRM domains in bacteria, at least here, and I think in archaea as well.
Okay.
So probably that maybe different type or different sequences, but yeah, as Miroslav said, I think he will be more familiar with the eukaryotic.
Okay.
But it's yeah, to be tested or to be seen more. We have more questions. So I wanted to ask about this specific complex, because there is this the problem of the system comes also from the very short RNA. Is this in the native system? This RNA is so short. I think, or for molecular dynamics, you just took part of the RNA that it's supposed to make a complex.
Okay, so these are experimental structures. So I took the RNA which was available in the experiment, which was resolved. Yeah, in case of HUR, we extended it a little. So yeah, the fact the behavior of the terminal interactions, the fact that the nucleotide is terminal here, it could slightly accelerate the problem, but yeah, because there is more dynamics, but it would happen eventually. Yeah, anytime the RNA in simulation separates from the protein, there is a big chance that it won't come back anymore and instead forms these RNA interactions. So in the systems where this happens later, then it might not happen on your simulation time scale. While in these systems where there is a lot of dynamics, it can happen very soon, but it will happen eventually. The force field simply has the global energy minima wrong.
Okay, I see. And since I don't see further questions, I think we can close, and we are running out of time. Thank you, Miroslav. I think that was very, very useful. The video will be uploaded at some point. And yeah, see you next year, and enjoy December, Christmas, winter, or palm trees, wherever you are. And see you next year.
Thanks. Bye.
Thank you.